subject might do a task. For example, here, a subject is opening and closing their eyes. They're typically in a magnet with using some stimulus, perhaps responding to the button box. And as we're doing this task, we're measuring the signals in their brain. And what we can see is the signals go up and down as they're doing this task. And we can map forms of statistical maps indicating where in the brain is most active, and make lots of sort of very informative conclusions from these types of studies. So I want to talk to you today about something that's a little different, which is, what if you just have a subject just lie in the scanner, okay, and really do nothing? Well, what happens when you do that, and you look at their brain patterns, is you can see their brain is fluctuating quite a bit. And if you just stare at this, you can sort of see it's not quite random. There are patterns that are reappearing over and over again. And in fact, if you look at any one part of the brain, it does look like a fairly random pattern. It looks just like noise. Okay? Remember, the subjects are really just lying in the scanner, either with their eyes closed, or we might ask them to have their eyes open, just looking at a crosshair. So these are just sort of the number of papers that have been published in fMRI, which is up to 3,000 cumulative, 3,000 per year. And if you look at resting state fMRI, right now, really, where resting state fMRI is really taking off. So over 700 papers published in the past year. This phenomenon was first discovered back in '94, and it really sort of languished for about 5 years. The original paper that sort of showed that resting state fMRI was useful was rejected 3 or 4 times, okay, before it's finally published. Okay? And so I'm going to sort of tell you the story about why sort of what's led to this huge sort of exponential growth in resting state fMRI. So here's the amazing finding. If you have someone tap their fingers and you map where in the brain their brain is active, you end up with a map of the motor cortices. Okay. Now let's say you just have the person lie in the scanner doing nothing. And you look at their signals, and you look and say the left motor cortex and the right motor cortex. These are the type of signals that you would see. And what you'll notice is they're highly correlated with each other. Okay? They're so correlated with each other that if you took the signal here in the left motor cortex and asked how is it similar or correlated with every other box in the brain, what you end up is a map like this, which looks very similar to this map here. So this was the amazing finding by Visual and others in 1995 that no one really believed for many years, but it turns out it's true. The field really took off. What it's all evolving is really marketing. You have to have the right marketing sort of employed to make the field take off. And it really took off when Mark Rabel sort of introduced this idea of the default mode network, which many of you have probably heard of. It's sort of the regions in the brain, primarily the posterior listening cortex, which have very high resting state metabolism, but tend to deactivate or have lower metabolism and lower blood flow whenever you're asked to do a task that requires your attention. So these red areas here are those areas that are correlated with the posterior cingulate cortex while the subject's resting. And so that forms what's called the default mode network. Now these areas in blue are indicating areas that are anti-correlated with that posterior cingulate cortex. And so if you look at these signals, so the orange is the default network going up and down, and sort of in opposition, the blue is what's called the task positive network or the attention network going in the opposite direction. So this was a really intuitive, nice picture of the brain, this sort of yin-yang of the brain, where either in your default network, you're sort of thinking about yourself, and then you switch to this task positive network where then you're actually maybe engaging with the outside world. So this has been one of the things that's been a really sort of fundamental idea in this area. Now, the really cool thing about resting state is it's so simple. Okay, and the subjects, so if you do have fMRI, often there's a lot of preparation. You've got to get the stimulus right, all the timings right. Resting state fMRI is great. Just put the person in the scanner, scan, you're done. Okay, which means it's really good for things where the subject can't actually even do a task. So we've heard a little bit about vegetative states and locked-in syndrome. So it's really a powerful paradigm for going into those populations that really can't actually do the task. This is showing that sort of widespread connectivity in areas for normal controls and much less connectivity in subjects with impaired awareness. And you can see here, there's a very nice correlation between these two sort of opposite sides, sort of interhemispheric connectivity here in this healthy subject. You can see it really falling apart in these vegetative subjects and impaired awareness subjects. And so it's a really powerful paradigm for going into these other populations. Another really taken off with resting state fMRI is its ability to sort of get a fingerprint of a person's brain, sort of a personality trait, in the sense that this is a video that we, from Howard Blaz's lab in the Institute for Neurocomputation here. And what you're seeing is the subject is sort of in a virtual reality environment. This is what the subject's seeing. And it's a 2-day experiment. On the first day, they're just randomly exploring this aircraft carrier environment. And on the second day, they're come back, and even though they weren't told really what to do, they're just sort of tested on how well they sort of can remember what they weren't actually told to remember. So it's a form of unsupervised learning where we're just sort of seeing, how well do people sort of absorb information about their environment, even if they're not told what to learn. And what's amazing is we then took 10 of these subjects and scanned them 6 months to a year later, just using a resting state paradigm. And what we found was that if you looked at the relative amplitude of their goal signal in the sort of the basal ganglia areas, you see an almost amazing correlation with the performance score. Okay, and so indicating that those subjects that have high goal amplitude in the basal ganglia will perform better than those that have low. Interestingly enough, also if you look at sort of connectivity, which say it's striatum and the posterior cingulate cortex, you end up with these amazing sort of correlations between sort of the performance score and the mean connectivity. So these are the types of findings that are getting people really excited about this in terms of whether you can sort of use these fairly simple measures that sort of predict performance. And this is just one example. There's other examples looking at working memory and other performance measures. Okay, the other thing you can do with resting state fMRI is you can apply a lot of interesting analysis approaches. So typically, one thing we'll do is we'll take a brain and we'll parcellate it into different anatomical regions. And then you can ask the question, how are different anatomical regions correlated with each other? Okay, so you take each ROI pair at a time, and then you would sort of enter in the correlation value into a matrix. And so this would be the correlation between, say, these two regions of interest. And then you just repeat that process over and over again for every ROI pair, or region of interest pair. And what you come up with is what's called a correlation matrix. Okay, so these are sort of matrices that you see a lot if you're in the resting state fMRI literature. And so each of these colors indicates how well one ROI or region is connected with another region. So from there, you can sort of go from this functional connectivity matrix, and then you can then plot it sort of in terms of an anatomical space or some other space, and sort of really sort of show a network diagram, essentially. This is really giving you the network information about how every brain region is connected to every other brain region. And so you can start doing what are called sort of network theory measurements on the brain. This is a study from Randy Buckner's group doing a very simple measure, which was looking at sort of what are the hubs in the brain. Okay, the hubs in the brain are those areas that have a lot of connections into them. Okay, so here, for example, the airline sort of analogy is, Chicago is a hub for United. All the flights go to Chicago, and if you're stuck in Chicago, there's no way to get out, right? So it turns out that one of the big hubs in the brain is the posterior cingulate cortex. Once again, that primary sort of brain region for the default mode network. And so this is sort of plotting those areas in red are the major hubs in the human brain. Interestingly enough, in this study, they also looked at amyloid deposition, and they found that those areas that were the hubs also tended to have the highest degree of amyloid deposition. You can take this network analysis to other sort of conditions. This is a study where they're once again looking at all the connections in the brain. And what they're looking for here is trying to figure out which connections are really distinguishing between a group of healthy controls versus a group with major depression. And so here they're just sort of plotting those connections in the brain in magenta, which tend to be highly predictive of who's going to be in a major depression state versus a healthy control. The other amazing thing you can do with this data is you can start taking a network and asking what are the modules in that network. So for example, here at UCSD, there's a network of all the faculty, and then the modules would tend to be different labs with tighter connections within a module than between modules. And so by doing that, you can start using this data to sort of parcellate the brain just from this data on its own. This is what's called functional parcellation. And you can just sort of ask, what are the modules of the brain? How's the data actually parcellating itself? And the amazing thing is if you do that, for example, this is a parcellation done by Cameron Paddock at the Child Mind Institute, where just from these resting state fluctuations alone, he's parcellated the brain into these functional regions, which look very similar to sort of the anatomical parcellation from, say, the heart box or apparatus. Okay, so we are really getting some very rich information from this, and there are now sort of methods where we can actually, this was done on sort of large sample subjects, where we are getting to the point now where we can stick someone to the scanner for 10 or 15 minutes and come up with a functional parcellation of their brain. The other amazing thing, this is a paper by Steve Smith in Kinetics 2009. This is sort of one name for this paper sort of is this is the paper that will end all fMRI. Okay, here's the reason why. What I'm showing you here are different networks. There are 8 different networks in the brain. These networks here are showing those areas that you would find to be networked just from a 10-minute resting state scan. I think they did about 10 subjects here. Okay, so just basically looking at the data and using the data to parcellate the brain into different brain regions. Okay. These networks here, shown on the right, are networks from like 30,000 task-related studies done over many, many years. So this is from the Brain Map Repository in San Antonio. And essentially, anything that shows up in a task-related, any network that shows up in a task-related paradigm is also showing up in the resting state paradigm. Okay. So once you have that, you can ask an interesting question, which was Steve Smith asked, is essentially every task-related paradigm you do, say it relates to say space and cognition, you can ask which networks does it recruit? Okay. So once you know these different networks and you have a table like this, it's simply looking up and saying, well, if I do any task, I know it's going to recruit these networks. Okay. So these are sort of just showing the power that in its resting state, the brain already sort of has all these functional networks in place that are then being recruited when you're asked to do a task. Okay, so this sort of method, this is one of the basic sort of methods that have really sort of led to this explosion of interest in what's called connectomics. Okay, which is has its history in the early days of neuroscience with these early diagrams from Hall, and then this sort of very famous visual network diagram from Van Esten. These are all painstakingly done from anatomical studies. Now we're at the point now where we can do this non-invasively. Okay, so this has led rise to what's called the Human Connectome project. Okay, so it's our field sort of analogy to the genome project. Okay. So this is the Human Connectome project, which is now into its just starting its 3rd year, and the idea is quite ambitious. They're scanning 1,200 subjects in St. Louis, 3 of them who are identical twins. And they're collecting resting state data on all these subjects. In addition, they're acquiring diffusion imaging, getting genetic information, and also doing some MEG scans, which I'll talk about later. So the amazing thing about this data, it's all going to be publicly available. Okay? The first 12 data sets have already been released and are available for download. They've also got some nice software that you can use to look at, to analyze the data. This is going to be and the data will be published basically as it's being acquired, sort of in chunks. So they're not going to wait till the end. The data now, you can actually start looking at the data and start playing around with it. So I think this is going to be a really important resource for our field. And what are some of the things that are really sort of making this project possible? One of the things that sort of happened in the last 5 years is the field has really pushed forward the technology in terms of how fast we can cover the brain. And what's made that possible is now, instead of just using, currently most studies maybe use a coil with 8 receivers. This is a coil with 32 receivers, and these are now sort of commercial versions of that coil. So modern MRI systems now use these very high channel count receivers, and by doing that, what we can do is instead of just going 1 slice at a time, we can go 4 slices at a time, 6 slices at a time, 8 slices at a time, and collect the data much, much faster. So for example, by doing this, the connectome is acquiring 8 slices simultaneously at a time. That's allowing us to do a 2 millimeter isotropic resolution at a TR of 720 milliseconds, okay, at a typical T for 3 Tesla. So we're going way faster than we typically go. So most of you are probably doing 3 or 4 millimeter resolution at a TR of 2 seconds. Here we're getting higher spatial resolution and we're going super fast. So what that allows us to do is we now have a problem. We have a lot of data, but it's also going to allow us to start looking at the data in a much more interesting fashion. This is a Steve Smith, a recent paper where he's looking at the temporal modes. And so essentially, we've talked about these brain networks, and it turns out it's not a big surprise, but these brain networks are not constant in time. They're coming in and out of play as you're doing different activities. And with this high temporal resolution data, we're going to be able to sort of really probe how is the brain switching between different networks. Another aspect which is not resting state fMRI, but is critical, is diffusion tensor imaging. And so this is looking at the diffusion of water in the brain, and most of you are probably familiar with these sort of amazing looking images that can be used to start tracing the anatomical connections in the brain. Now, I put this slide in for Jim Brewer, but I don't see. Jim, are you here? All right, okay. So right after you said I had to talk about this, I put this in my talk. Okay, so one of the key things is with these new technologies, we are tending to have what's called a longer readout lane. So there is much more distortion typically. So here you see distortion where the brain is really pulled out this way. If we change what's called the phasing cogradient, then the brain is squished into this way. So the nice thing is, using a method sort of introduced by E.S. Rodgers in 2003, if you acquire both these directions, you can use that information to create what's called a correct image. And this is going to be sort of the method used in the Connectome project, or one of the methods. It is now released as part of the FSL package as the top-up, so E.S. Rodgers has implemented it as top-up, and so this is something that we'll be making available as part of some of our packages, and so I encourage some of you to sort of look into this. This is going to be a very nice technology to use going forward. The other thing the Human Connectome project is going to be looking at is not just resting state fMRI, but also looking at other metrics such as MEG or encephalography. And MEG has much better temporal resolution. It's in the millisecond range versus functional MRI, but it's typically had much poorer spatial resolution. So instead of being at the millimeter resolution, we're really talking about being at centimeter resolution for MEG and EEG. But it turns out that despite that, at least for these large-scale networks, the networks that we see with fMRI, shown here on top, we can also start seeing with MEG. Okay. And why would we want to sort of add MEG to the arsenal? Well, one reason is because the bold signal that we use for fMRI still has a lot of issues in terms of we don't know whether it's really a neural signal or whether it's affected by vascular compounds or metabolic compounds. The other thing is that the temporal resolution of the MEG is much better, and so there's a question of whether we can take advantage of that. So this is just some recent data from our lab showing connectivity maps with MEG and bold in different conditions. And the main thing is, here's the subject, they were before they took caffeine, here's the subject after caffeine. And you can sort of just see visually that the amount of connectivity shown by the MEG, the decrease here, is very similar to what we're seeing with the fMRI. So really sort of showing that we can start using these two modalities to see similar things. Now the advantage of MEG is that we can go further in and actually look at what sort of bands are really driving that change. And so here we're seeing really the alpha and beta bands are really driving sort of this decrease in connectivity. And so we start understanding a little further what's actually happening in the brain. The other sort of modality we have at our access is, and it's really well suited for resting state fMRI, is simultaneous EEG fMRI, in which we scan a subject and at the same time we actually measure their EEG signals. This is another area that's really sort of blossomed and I think is going to continue to grow in the coming years. The problem is this is the EEG signal you get in the scan. Okay? For those of you who know EEGs are not like spindles or K complexes, this is gradient noise. Okay. The good news is a lot of these problems have been solved. And so there are algorithms now for going from eliminated most of the noise, which is due to the gradient switching on and off. We still have some residual noise. First, notice this sort of 1 second period here, and that's related to the heart beating in the magnetic environment, causing even the small motions of the wires on your head as your skull pulsates is enough to cause this residual artifact. The good news here again is that we can largely take it out and get some very nice, clean EEG data in the magnetic environment. So this is really nice because then we can start sort of parsing out what's happening when we see these things in the fMRI. These are three different, these are the connectivity maps showing the default network in 3 different subjects. But you notice there's quite a lot of variation. This is quite widespread, a lot of red here, and much less here. Here you're actually seeing a much nicer delineation of sort of the default network in red and task positive network in blue. Here, not so much. With the EEG fMRI, we start asking, well, what's different between these subjects? And it turns out, if you look at the EEG spectrum, this subject here has a very high altitude, indicating a very high level of vigilance with their eyes closed. This subject here, who's sort of in the middle, is starting to get less vigilant. And this subject here, who has a lot of this sort of widespread, what we call global signal, is very low level of vigilance. And so we are starting to understand sort of the connection between these EEG sort of states and also the fMRI signal. Sarah talked a lot about sleep, and resting state fMRI is perfect for looking at sleep. This is a paper that just came out where they essentially used the EEG to look at different sleep stages. Okay. And then what they did was they had an fMRI at the same time, and they looked at the connectivity between different brain regions, and then trained a classifier, a support vector machine, to predict what the sleep stage was. And this was the fMRI prediction. These are just showing that, for example, when you use the manual sleep staging or the automatic sleep staging, that you get very similar. You can identify wake sleep. The spectra from these different predictions are all very similar. Okay? So I think this is a really nice application where we're really going to be able to sort of better get a better handle on sleep by using sort of the differences in brain connectivity during sleep stages and correlating that with different EEG signatures. Now, I've talked a lot about, so the Human Connectome is going to involve a lot of this, and that data will be available. Now, at the same time, what I want to point out is I think the other really good thing that resting state fMRI has brought to our community is sort of a different culture. Many of us collect our data, we hoard it, protect it very carefully. The resting state thing has a really different culture. It's really about sharing. It's our summer of sharing. Okay, so this, you probably don't know that this was the summer of sharing. Okay, so if you share any data, you can share next year. So if you look online, just Google Functional Connectomes project. There has already been a lot of people that have already shared their data. And there's a real culture towards sharing either data, or if you don't want to share your data, share some tools. Just share. Okay? Because it'll benefit everyone. And the nice thing is, the thing that makes it possible is resting state fMRI doesn't require, anyone can do it. And we're pretty much all doing the same thing. Okay? The Human Connectome project will be able for everyone to analyze. 1,000 Connectomes, there's already over 1,500 data sets available for anyone to download here at the 1,000 Connectomes project. And it's led to work like this, for example, where Nora Volko's group at NIDA looked at sort of hubs in the brains, and this is just comparing from all these different sites, from Beijing to Queensland to Taipei to Palo Alto, sort of how similar are connectivity patterns from these different studies. So I think in closing, I think we are really at the cusp of sort of a paradigm change in how we do neuroimaging. We started off with classical maps at the beginning, at the early stages of neuroscience. MRI has definitely changed how we think about it, both MRI and fMRI. And now you could argue this is a sort of behaviorized block here. We're really now engaged with connectomes, where we're looking at brain networks, how they differ between disease and health, how they can predict performance, and how these brain networks change over time. So I think it's a lot of information. I mean, once you start looking at the connection between every part of the brain with every other part of the brain, and also asking how do those connections change over time, you've got a huge amount of data to look at. And I think that's going to be really important for us to all keep in mind that as we start trying to understand and unravel the mysteries of the brain. So I'd like to thank you for your attention and thank my collaborators and the group.